Hello everyone, welcome to BrainMap. This seminar series is co-sponsored by the P41 funded Center for Mesoscale Mapping housed in the Martinos Center. It is our great pleasure today to have Dr. Jae Mo Park, and thankfully our colleague Ethan volunteered to introduce him to the audience, so I will hand it over to her. Ethan?
Great, thank you. Yeah, thanks for the organizer. Do you have other announcement to make before I introduce? No? Okay. Okay, great. It's my great pleasure to introduce our speaker today, Dr. Jae Mo Park. Jae Mo is an expert and has made major contribution in the development of non-invasive MR imaging techniques to study the in vivo metabolism and function in disease and normal subjects. And he has investigated in vivo metabolism using hyperpolarized C13 MR spectroscopy imaging. And he also developed advanced MR techniques to study tissue characteristics. They are now doing a clinical translation of metabolic imaging in human in clinical trials to study the metabolic and functional imaging of brain, heart, liver, and skeletal muscle. There are many ongoing clinical trials at the UT Southwestern Medical Center. Jae Mo received his master's degree from Electrical Engineering Department at Stanford University. And following that, he got his PhD also at Stanford. And that's where we overlapped. I was there at the time working for GE Healthcare, and I was collaborating with Jae Mo in many of the very interesting hyperpolarized C13 imaging study early on. And now Jae Mo is an associate professor at UT Southwestern Medical Center, Department of Biomedical Engineering, as well as Department of Radiology. So welcome, Jae Mo, to give us this BrainMap presentation. Thank you.
All right, thank you very much for the introduction, and also thank you for the organizers for inviting me. So today I'm going to talk about hyperpolarized bicarbonate as imaging biomarker of neural metabolism and function. So I'm going to very briefly talk about hyperpolarization technique, because I assume that because Ethan is there and other people who are working on the hyperpolarization is in MGH. So I don't I assume that a lot of the audience in MGH already knows some background, but just in case, I included a couple of slides. But I'm going to show that I'm going to talk about it later. For now, let me start with some very basic. So probably I'm the novice here, but in brain metabolism, glucose and glycolysis has been the main substrate and metabolic pathway for a long time. And for a really conventionally, glucose has been the primary substrate in the brain. And also, for a long time, astrocyte neuron lactate shuttle has been the dominant theory for understanding cerebral energy metabolism. So on the right side, this one is actually, I borrowed from this journal. So not all the metabolic pathways are accurate. The regulatory signaling pathways not accurate, but this one actually gives a good overview about the metabolic interaction between astrocyte and neurons, and also showing the glucose, how the glucose is metabolized, and then connected to neuron. So basically, according to astrocyte neuron lactate shuttle theory, the glycolysis in astrocyte is primary starting metabolic pathway for glucose. So glucose is primarily uptaken by astrocyte, and the astrocyte breaks down glucose to pyruvate through glycolysis. During the process, the glucose can be also stored as a glycogen, and when the glucose is not readily available, the glycogen can be also broken down to through the lower part of the glycolysis to generate pyruvate. And once pyruvate is generated in astrocyte, instead of going through the mitochondria, which is more energy efficient metabolic pathway, this one is converted to lactate, and then lactate is released through the MCT1 or MCT4, and then this lactate produced from astrocyte is transferred to neuron, and then the neuron is uptakes lactate through MCT2, and then converted back to pyruvate, and then pyruvate is further metabolized in the mitochondria, mostly through the oxidative phosphorylation in the T-phase cycle. So, so this is like a primary, like a dominant metabolic pathway according to the astrocyte neuron lactate shuttle theory. Glucose is converted to lactate in astrocyte, and in neuron, lactate is also going forwarded into the mitochondria. So a lot of neurological disorders, glycolysis in astrocyte and the oxidative phosphorylation in neurons have been the major focus. Over the last years, like more recently, the lactate has been getting more attention as a substrate. So as I mentioned, according to the previous theory, the lactate is generated from astrocyte, but this more like an emerging theory, which is called like a lactate shuttle theory, is actually using lactate as a substrate, not as a product from astrocyte, but lactate is circulating in the body, and then this circulating lactate, which is not necessarily from generated from astrocyte, can diffuse into the extracellular space from the blood plasma, and then can be further metabolized in the neurons. So this actually explains a couple of things. One is lactate. A lot of this one is one example. Several studies actually reported that the brain cells prefer lactate over glucose as an energy source. And and another thing is there's some energy balance was not really matching between glycolysis and the oxidative phosphorylation, but by putting this lactate in between as a vehicle that links these glycolytic and oxidative metabolism, it really makes sense. So so in more contemporary the cerebral energy metabolism concept is this neuron is actually can metabolize multiple substrates. So previously, like conventionally, like glucose and glycogen are the primary. The lactate was generated from the astrocyte, but now lactate can be directly diffused from the blood, circulating blood. So so either this one is from glucose, glycogen, or lactate. To further, metabolize in the mitochondria in the T-phase cycle in neuron, it should go through pyruvate. So the pyruvate is more like in the important, junction for the multiple metabolic pathways. So and there is a couple of important, steps for pyruvate is linked to the T-phase cycle. The one is the mitochondrial pyruvate carrier. So pyruvate is carried into the inner mitochondria through MPC, And this, once pyruvate is entered into the mitochondria, can be further metabolized through the pyruvate dehydrogenase. This is irreversible metabolic pathway. And then once pyruvate is converted to acetyl-CoA, this one is primarily, although it has a couple of other metabolic pathways, but primarily this one is directly going into the TSA cycle. So measuring PDH, so the PDH is kind of regulating enzyme and entry enzyme for pyruvate to the mitochondria. So like understanding PDH and measuring the PDH flux is quite important for cerebral energy metabolism. So pyruvate is located here. So like I said, either glucose or glycogen or lactate, or even small portion of glucose and glycogen going through directly through the neuron. It should go through the pyruvate. And the first enzyme that primarily regulates pyruvate oxidation is the PDH, which is located here. So here is, that was the major motivation of using hyperpolarized pyruvate. So because hyperpolarization allows the carbon-13 MR spectroscopy or imaging or spectroscopic imaging within a minute, usually like a maximum like couple of minutes, for various reasons. So what is hyperpolarization? The hyperpolarization is creating nuclear spin polarization much greater, much greater than that can be achieved at normal thermal equilibrium. So this is like a polarization term, which is proportional to the the MR signal is proportional to the polarization term here, P. And this P is also proportional to the population difference between these two energy level divided by the total, the population. So like a relatively like small number of population difference under a thermal equilibrium, but once you make it hyperpolarized, the population difference is much bigger. So you may have heard about polarization percent. So the polarization percent means how much population difference compared to the total number of population. So if you have like 100%, there's nothing in one energy level compared to the other. So how low is it in thermal equilibrium? So at 3 Tesla for carbon-13, at room temperature, polarization level is this low, 0.00026%. And then these are the two polarizers that we have here at UT Southwestern. I believe MGH also has two of these spin aligners. So these are state-of-the-art preclinical and clinical polarizers, DMP polarizers. And based on our measurements, right after dissolution, we get about 60 to 65% polarization from spin aligner, and then from GE Spin Lab, we get about 35 to 50% polarization. So if you compare these two, the actual amplification factor is about more than 200,000 for the spin aligner, and for the spin lab is more than 100,000 times at 3 Tesla. So what can we do? We can do kind of in vivo enzyme assay. So in enzyme assay, if you have done any enzyme assay, you prepare substrate, you prepare some tissue sample or cell culture, and then there are enzymes in the cells or in the tissue, and then you provide enough substrate and coenzymes, and then you measure the amount of product. So you're measuring the enzyme, the maximum enzyme activity you can get through the enzyme assay. In vivo, you don't have to add these substrate and coenzymes because is already in the body, and then all you have to do is inject the substrate. So, and then you can measure the product. So if you are injecting pyruvate, you can expect that this one can be converted to lactate through lactate dehydrogenase. And another pathway is the pyruvate dehydrogenase that I mentioned. This is the entry enzyme to the mitochondria. And during the process, you can, like, depending on the labeling position, you can detect bicarbonate production, which is actually equilibrium with carbon dioxide, which comes out when pyruvate converts to acetyl-CoA, or you can detect the carbon-13 labeled lactate. So this is like the patient data published like a few years ago from Cambridge group, and this is like a human patient with breast cancer, and then you can see a large amount of lactate production in the tumor area. And this is, our data acquired at the Southwestern myocardium, in the long axis plane, so you can actually see the myocardium from the human heart. So everybody is excited about this. So what can we do with hypopolarized C1 label pyruvate? So like a C1 label pyruvate is, the carbon-13, the carbon in the carboxyl group is labeled with a carbon-13. And this is actually a pretty good choice because the T1 relaxation time is actually longer. So you can actually have a longer observation time. And the C2, a C3 is really short. It has a really short T1, and the C2 has additional coupling, proton coupling. So you actually has a splitted peak and also shorter relaxation times. So if we inject a C1 label pyruvate, this one is going to go through the monocarboxylate transporter, like lactate, and then it can be converted to C1 lactate, and also it can be further, metabolized in the mitochondria. So as I explained, the blue, the pyruvate can, a labeled carbon can be decarboxylated as a carbon dioxide, which is equilibrium, makes equilibrium with the bicarbonate. The blue fonted, metabolites are the carbon-13 labeled products, and therefore you can actually detect the signal using MR. So in neuron, so if you can actually see pyruvate metabolism, you can actually estimate, maybe like have a good idea whether glucose lactate or glycogen is actually oxidized in the mitochondria. So the production of bicarbonate, which is produced through the PDH flux, indicates PDH flux and pyruvate oxidation. So this hypopolarized carbon-13 pyruvate is also different from other imaging modalities. These are probably the other two common imaging methodology for assessing this glucose metabolism. So as I mentioned, hypopolarized pyruvate detects the pyruvate oxidation either from glucose, lactate, or glycogen. So you can actually measure the entire big picture of the cerebral metabolism. FDG PET is more like a detecting glucose uptake because once FDG is uptaken, it doesn't further metabolize through the glycolysis. So you are actually measuring the glucose uptake. Deuterated glucose or deuterated metabolic imaging method is detecting glucose oxidation. Including glycolysis and glucose oxidation. So these are the major differences. So my questions, so I've been working on this one for a while, and I had these questions several years ago. So the first question is hypopolarized bicarbonate primarily from neurons? And the second question that I had was, is hypopolarized bicarbonate directly from hypopolarized C1 pyruvate, or it can be converted to something else and then generating bicarbonate. The third question is, can we detect neuronal activation by monitoring hypopolarized bicarbonate production in the brain? And the last question that I am going to talk about today is, does hypopolarized bicarbonate reflect altered cerebral metabolism in neurological disorders? So I'm going to give a couple of examples. So let's talk about the first one. So is, hypopolarized bicarbonate from neurons? So we don't have a direct answer for that, but I found some of the answers from the literature. So the contribution of neuronal metabolism to hypopolarized bicarbonate, I could get a pretty good understanding from numerous, the literature. One of them is, actually reporting that the relative lactate transport capacity of neurons and astroglia can exceed glucose oxidation rates when they are provided with external lactate, and then it can actually create a shared lactate. So this one is well aligned with the lactate diffuse theory. And this one was discovered by infusing carbon-13 labeled lactate without hypopolarization. And from the same study at supraphysiological plasma lactate, like hypopolarized lactate case, or hypopolarized pyruvate case, the contribution of plasma lactate to brain metabolism through direct lactate transport increases up to like 60%. And more importantly, lactate permeability is much smaller in astrocytes than neurons when lactate is injected intravenously. So if you are injecting, hypopolarized pyruvate or lactate, the lactate or pyruvate permeability to astrocyte is much, lower than neurons. So it's more likely gonna deliver, transported into the neurons. So, I mean, it's not a complete answer, but we have more data, but it's not complete, but we have more ex vivo data that support probably this, the hypopolarized bicarbonate is primarily from the neurons rather than, astrocytes or other microglia or other brain cells. So the title could have been hyperpolarized bicarbonate imaging biomarker of neuronal metabolism instead of neural metabolism. But I just wanted to be cautious in terms of the title. But I'm more convinced that this is bicarbonate is from primarily from neurons. And the second question is hypopolarized bicarbonate directly from hypopolarized C1? So what does it mean? So so we did some pathway analysis, of this bicarbonate production. So because if you are injecting hypopolarized pyruvate, as I explained, it can go through the intracellular cytosolic space and then can be further metabolized into the mitochondria. But alternatively, it can be converted to lactate before actually entering to the cell. And so if it does that, lactate can enter into the cellular space and then convert it back to pyruvate and going into the TCA cycle. So either this way or either that way, it's gonna create bicarbonate. So how much is it gonna be? So I thought like maybe this is dominant way, but it turned out this one is actually very substantial. So why is that? Because the red blood cells in the blood, circulating blood, has red blood cells. Red blood cells don't have mitochondria, so they don't have oxidative phosphorylation, but they have lactate dehydrogenase. So you can actually create lactate in the bloodstream. Other possibility is other organs, so like a liver. So you can some other studies. I think the first paper actually reported that by saturating lactate signal from the liver, you can actually suppress a lot of lactate signal in the blood. So other organs also can contribute lactate production. So once lactate is produced, um, the redox state is more favorable for to be lactate. So lactate can be circulating and then go into the cell in the brain and for the oxidative metabolism. So we tried to mimic this hyperpolarized pyruvate study, and also we tried to compare with a lactate study. So we injected a uniformly labeled pyruvate and also uniformly labeled lactate without hyperpolarization when we injected into rats. And then we performed the carbon-13 NMR isotopomer analysis to compare of the brain tissue. We collected a couple of minutes after the bolus injection, and to compare the metabolic phase of the pyruvate and lactate following this bolus injection. And the harvest tissue actually showed almost identical spectrum. So we collected about I think 5 or 6 animals, and then we couldn't identify any significant difference between these two labeling patterns. So that indicates that the similar amount of carbon-13 is passing through PDH between the pyruvate and lactate injection based on primarily from the C1 succinate and C6 citrate. But other like glutamate and glutamine patterns were also very similar between these two comparisons. Another experiment we did with humans. So we wanted to see how much lactate we can are actually contributing to the bicarbonate production in the brain. So we made 2 different R pulses. Like this one is spatially selective. So this one is single slice excitation, and this is the spectral profile. So x-axis is the frequency, and as you can see, pyruvate is excited for 5 degrees, and then bicarbonate was 90 degrees, and the red one, RF1, is exciting 10 degrees, and the blue one is exciting 90 degrees. So by comparing these two, and then we acquired like a dynamic spectroscopy from a human brain, right after injection of hypopolarized C1 labeled pyruvate. So by comparing these two, you can actually, if there is any bicarbonate production difference between this RF1 and RF2, that indicates that the large portion of, some portion of bicarbonate is coming from lactate. So we did it, and then we actually detected, so when we were using small flip angle for lactate, the bicarbonate production was almost doubled, and lactate production was also decreased because if you're using smaller flip angle, lactate production decreased because we're using small flip angle, but bicarbonate was like doubled. So this one indicates that the lactate contribution, even if you are injecting hypopolarized pyruvate, but lactate is already substantially generated before arriving to the brain or right after arriving to the brain. So so we concluded that from the second question, we concluded that hyperpolarized bicarbonate can be directly from hypopolarized pyruvate, but at the same time indirectly from hypopolarized lactate. And the third question is, can we detect neuronal activation by monitoring hypopolarized bicarbonate? So based on these two, we have a good reasons to look at and assume that the hypopolarized bicarbonate is reflecting the pyruvate oxidative metabolisms in neurons. So can we actually detect this neuronal activation by monitoring this bicarbonate? So assessing the PDH flux through hypopolarized bicarbonate has several advantages for over conventional imaging approaches. So the first advantage is really quick because we don't have a lot of time. So all the observation is actually pretty quick, and the signal to noise ratio is really high. So we have a pretty good sensitivity. And the second advantage is, we are actually detecting PDH activation directly. As you can see here, there is a series of physiological metabolic processes involved in brain activation, and the increased oxidative metabolism, demands a continuous supply of oxygen and glucose to neurons, and also astrocyte, and this collectively leads to an increase in CVF and CVV and metabolic rate of oxygen consumption. And this combination of changes actually form the basis of both fMRI. So as you all know, both signal is from the deoxygenated hemoglobin, which is highly paramagnetic, and then we are detecting a T2 star perturbation in MRI. But this one is multiple steps away from actual metabolic changes. So this one is more like indirect way to detect. So some people are really criticize this both fMRI method because it's not really like a direct detection method. And one of the well-known disadvantage of both fMRI is it has vascular delay and during the signal acquisition, and so it can be underestimating the level of neural activities. So and also neurovascular coupling is not also an issue because when there's a metabolic rate goes up, there's a more perfusion, and then decoupling these two is gonna be the big challenge. So, but in contrast, if you're detecting hypopolarized bicarbonate, the PDH activation, so PDH activation is a direct measure of neuronal activity, so which is less susceptible to the neurovascular coupling, and also less affected by the blood oxygen level. So we tried, but before we actually tried, we noticed that bicarbonate signal in general is much lower than pyruvate and lactate. So before actually doing that, we did a lot of brainstorming how to reliably detect this bicarbonate signal, like meaningfully. So one idea was actually, instead of let's give up the imaging. So these are actually phantom data, carbon-13 labeled bicarbonate phantom. And then we tried various voxel sizes. These are all with 90-degree excitation. So as you know, voxel size, the bigger the voxel size is, the SNR goes higher. So, when you change these, SNR, the peak SNR was from like 13 to like 19. But if you actually give up this and then try to measure the spectroscopy, even with a small flip angle, this one was acquired with a 5-degree excitation. But you get much higher signal-to-noise ratio. This is actually coming from one channel of 8 carbon-13 channels in this carbon-13 proton head coil. So this one is showing the channel combined the carbon-13 signal across the phantom. And this one is coming from the only one channel. And as you can see, if you are actually reducing, if you're selectively looking at each channel, you can get pretty much the signal from the close nearby brain tissues. So you can get some kind of like a regional spatial information by looking at each channel signal. So but this one is really reliable compared to the actual imaging approach. So we applied this one for the activation study. And as I mentioned, saturating lactate is going to also decrease the bicarbonate signal, and also it also not detecting lactate oxidation directly from lactate into the oxidative phosphorylation. So instead of using 90-degree excitation on lactate, we use a small flip angle on lactate, the red one, for the animal study. And also, we used circular, conventional, like a blue-yellow checkerboards that alternate with like a black screen with a cross at the center. So it's alternating, and then for 20 seconds, and then showing black screen for 20 seconds, and then move back and forth during the acquisition. So by doing so, so this one is visual stimulation. So we could detect visual cortex bicarbonate signal by only looking at channel 2 and channel 3. And the red yellow signal, this is actually a bold fMRI signal collected from the proton RF coil, because the coil was actually proton and carbon-13 dual frequency. And this is the results. So this one is time averaged signal. And the one thing you need to notice, aware, is the pyruvate was excited with 5-degree, the lactate was excited with 10-degree, and bicarbonate was excited with 90-degree. But stimulation, with a visual stimulation, we could see more bicarbonate signal. Although it's very difficult to quantify because of the different flip angles were applied. But we thought like maybe for the proof of concept, this is good enough. And the lactate was not actually changing very much. Maybe this is because of the small flip angle, 10-degree flip angle. And these are the data from 7 subjects we collected. So this one is resting, and this is stimulation, bicarbonate change, but this one is normalized to the sum of all 3. Although this one is not really good metric, but we didn't have any polarization level. So this is probably the best we could analyze. So with the stimulation, bicarbonate was consistently produced more. So, and then later, we also, thought about, is it really affecting, increased, pyruvate oxidation in the brain? Because, a couple of papers actually reported that, the pyruvate, monocarboxylate transporter pyruvate transport into the cell, intracellular space, or through the BBB is a rate limiting step. So if this one is rate limiting step, you know, even if we have like a, activated PDH, we may not be able to detect the increased bicarbonate. And then, we also did the animal study by, opening BBB, and then it actually increased the bicarbonate production in animals. So this one is actually showing, so like gadolinium. So after applying, focus ultrasound to open the BBB, so you can actually see the BBB opening through the gadolinium enhanced, T1 weighted MRI. And then this is the overlay bicarbonate signal. So as you can see, bicarbonate signal went up much higher. And all the animals we applied this actually showed exactly like a matching spatial information. This one actually showed the biggest difference. So this one is, gave me more, the puzzling analysis or interpretation of these. But I also learned that if PDH is elevated by brain activation, it also increases the uptake of lactate. So lactate uptake is actually shown by several groups. And then so we understood that maybe what we are seeing is real, and the bicarbonate signal is increased bicarbonate signal actually reflects the upregulated pH flux by brain activation. And also we found the ex vivo study. It was done by the Israel group, and this paper actually investigated the association between the neuronal activity and the cellular metabolism using hyperpolarized signal label pyruvate ex vivo. So they tried to detect the hypopolarized bicarbonate in rat brain by applying two different drugs. One was tetrodotoxin, which is blocking the action potential, and the other drug was oligomycin, which is which blocks also active action potential generation. And then they could detect like the second drug is actually inhibiting ATP synthetase. And by using this drug, they could see dramatic difference in the bicarbonate production, which is reflecting, indicating that the neuronal activity, the amount of hypopolarized bicarbonate is related to the neuronal firing. And the last question is, does hypopolarized bicarbonate reflect altered brain metabolism in neurological disorders? I'm going to show like a couple of examples. So we try to detect the, the brain, the metabolic change, pyruvate metabolism changes over time, right after mild acute brain injury to rat brain. So this, we use the control cortical impact on the right side of the brain. And this one is like, we measure the 2-hour, 1 day, 2 day, 5 day, and 10 day longitudinal, which was really challenging, but we tried like 30 different rats, and then eventually we completed the data, and then this is the results. So we also tried to measure the contrast from the proton T2 weighted MRI. And as you can see, 2 hours after, the contrast almost like immediately showed up and then gradually went down. And we also tried to measure the bicarbonate contrast. So 2 hours later, the metabolic change was not very significant. But gradually, 1 day and 2 days, their metabolic change was much stronger, and even 5 days, and then eventually we went down. Lactate contrast was smaller, but there are other two groups have also reported measured the lactate contrast. I mean, they reported they reported lactate contrast is very obvious, but we didn't see a lot of lactate contrast. It's like a 5 to 10% change, but bicarbonate was like up to 30%, or some of them actually shows 50% difference. So like a bicarbonate is more sensitive, was more sensitive, and also it showed a little bit delayed metabolic response to the primary injury in the rat brain. So we also noticed that some of the rats were keep showing increasing bicarbonate signal in the normal side of the brain. And these are the four, it was not very consistent, but some of them were pretty obvious. So these are the four rats that we compared. This is bicarbonate image at day 1, and this is day 2. This is normalized to the total carbon signal. And as you can see, it's much more bicarbonate was coming produced in the normal side of the brain. And this one is also showing the normal side, and then this one too. And these are the highest bicarbonate change, the 4 rats with the highest bicarbonate change in the normal side of the brain. And, there must be, the more, there must be some mechanism. And one of the possible, theory is, after the injury, the astrocyte activation happens, and then, the glycolysis goes up. So it's called hyperglycolysis, and the neurons are dying, and the elevated lactate production is, accumulating in the extracellular space, and then it could be moved to the normal side of the brain, and then it's metabolized in the neuron, healthy neuron, and then creating more bicarbonate signal. So we are actually doing more ex vivo study right now. Hopefully we can get reasonable conclusion from that. And we also tried in humans. So this one is a healthy human brain, and this is pyruvate, lactate, and bicarbonate. As you can see, this one is from the spectrum, not using the spectrally selective excitation and imaging method. So this one, I think this is more accurate. And this page, we collected about 10, 11 patients with the TBI. And this patient showed had a motor vehicle car accident, and we recruited him 3 days after, and then he had a loss of consciousness after the injury, and then airbag deployed, and he didn't remember where he got impact, but his bicarbonate image was much lower in one side of the hemisphere compared to the other. The lactate and pyruvate were kind of normal. And this is another subject. We recruited him 1 day after injury on the left side of the brain. The brain itself was completely okay, but he had a pretty obvious injury externally, and then also contusion and edema as well. And then as you can see, the bicarbonate signal was almost disappeared. At the same time, the other side of the brain was also bicarbonate signal was decreased. So we have about 4 subjects that actually show similar behavior. So we think that because these are a close skull injury, so when you are hitting, you have an impact, like you have a coup counter coup injury from the other side. Maybe this is what we are seeing. More recently we started recruiting professional fighters. So these are the fighters with mixed martial arts, and then we recruited them. So far we recruited 5 subjects, and these are showing, and we also compared lactate and bicarbonate. And the top row is showing 1 year, more like a young athlete with 1 year of career, professional career. And the bottom row is from a little older, more experienced athlete with 14 years of professional career. And we are kind of surprised that because, bicarbonate signal was completely messed up in this person, although, we probably have to recruit more people to generalize this observation. But, after 1 year, bicarbonate signal was pretty good, but, 14-year experienced MMA fighter didn't show much of bicarbonate. So we recruited back about 4 or 5 months after, and then we also still got similar results. And this is the final data that I'm going to show. So we also are doing patients with gliomas. This one is showing, high glioma, glioblastoma patient here. And then the pyruvate perfusion was higher and the lactate signal was higher, but bicarbonate signal was completely disappeared around the tumor. This is another example with a high-grade glioblastoma. As you can see, lactate signal was elevated, but bicarbonate signal was disappeared. We also, recruited some low-grade glioma patients, and as expected, lactate signal didn't go up around the tumor. Bicarbonate is difficult to tell. But one thing that we noticed later, they didn't know either, but later he had small tumor in the posterior side. And then one thing we noticed that bicarbonate signal was missing in that area. And right after this one, we did another injection and then to detect the sagittal images. And one from here, one from the other side. And then the tumor area also didn't show up any bicarbonate signal, although the normal tissue was like showing some bicarbonate signal. So we think that this might be high grade or more aggressive tumor, not the low grade. But it's not confirmed because they didn't do any biopsy in this tumor area yet. So in summary, hyperpolarized pyruvate probes pyruvate oxidation by detecting hyperpolarized bicarbonate in the brain. And this bicarbonate production, pyruvate oxidation means, the glucose, the substrate could be from glucose, glycogen, or lactate. And to answer my 4 questions that I presented earlier, the hyperpolarized bicarbonate is primarily from neurons and can be produced from pyruvate directly and also lactate indirectly. And the bicarbonate is sensitive to several PDH activation and also altered in TBI and GBM, and also probably other neurological disorders. So I'd like to thank my collaborators and also our center support and my lab members who are usually very happy like this, and also the funding support. Thank you.